We're group SKH, and our gene is the Bradi1g25180, which encodes for a G-type S-Locus receptor-like kinase. The main goal of our research is to determine the function of this gene in Brachypodium distachyon, which is the plant we've been working with. We used three major approaches to answer this research question. First, we used a gene model predictions, specifically a homology-based model. Next, we used mutant analysis. And lastly, we looked at gene expression analysis, both with existing data and a novel experimental design. Reverse genetics is a pretty popular tool to determine gene function. Essentially, it involves taking a genomic sequence and breaking it with a mutation, and then seeing what kind of phenotypic effects happen as a result. This is a model of our gene, and we know that it's a good model because it's corroborated by RNA-seq data.
We used phytosome and NCBI domain searches to identify functional domains in our protein. Both databases classified our protein that our gene encoded as a G-type S-Locus lectin receptor-like kinase. Even without having a structure of the protein in its native conformation, we could tell that it was probably membrane spanning because there was a receptor region near the N-terminus and a kinase near the C-terminus. And that usually indicates that it's a receptor kinase which spans the membrane. It also told us that this protein was likely involved in phosphorylation of substrates and maybe signaling pathways. So knowing the classification helped direct our literature search, and we found that when G-type LEC RLKs include the S-Locus, which ours did, they usually are involved in the self-incompatibility response since the S-Locus is involved in self and non-self recognition. So we decided that our protein was likely involved in a pathway response similar to or the same to the self-incompatibility signaling pathway of this Arabidopsis G-type S-Locus lectin receptor-like kinase, shown here on the left. This is a model of the functional domains of the protein in Arabidopsis, as you can see on the right, and they are the same ones in the same order found in the protein encoded by our gene. And the sequence itself, not just the domains, is very similar to Arabidopsis G-type receptor-like kinases, as you can see in this phylogenetic analysis.
Next, we wanted to confirm that our mutant plant did in fact contain the mutation that we were expecting it to. So with Sanger sequencing, we confirmed that our mutant plant had a guanine to thymine substitution, resulting in a stop codon. So this protein in the mutant plant would be truncated and therefore would not have any functionality. We compared the wild type and mutant sections from lower peduncle, stained for the organs and the vasculature of the stems. Just from these images, we can see that it seems like the mutant might have decreased cell thickness, cell wall thickness, but due to the lack of the right analytical tools, we were unable to quantify this thickness, so we have inconclusive results from the staining and microscopy. Next, looking at gene expression data, we see that our gene is highly expressed in stems and the internode regions, both in young and adult plants. Our gene is also highly expressed in young seedlings. We also found that it followed a thermocycle where it's highly expressed in cold environments and expression is decreased in warmer environments. We wanted to further investigate this thermocycle regulation by conducting statistical analyses, and we found that gene expression levels are higher in cold treatment compared to controls, and these differences we also found were significant.
So this gene expression data was kind of important for us realizing that our hypothesis might not be right. So relative to expression in other tissues, the expression of our gene in the anther and pistil regions is very low. And this isn't what you'd expect to see for a protein implicated in the self-pollination response. As it turns out, it's actually not. And this is because Brachypodium is self-fertile. It doesn't have a problem with self-incompatibility. It doesn't reject self-pollen like we assumed it did. So it probably wasn't involved in this pathway like we previously thought. To design our experiment, we searched for some more possible functions for G-type LEC-RLKs, and there were a lot. Here's just a couple of examples out of dozens. We wanted to see if our gene was involved in abiotic stress response in plant growth and development. So our experiment measured differential expression of our gene in the presence of an abiotic stressor that would stunt plant growth, caffeine. We studied expression in young seedlings. We did this because our gene is known to be highly expressed in young seedlings, and RNA extraction is just easier with young tissue. So we grew the seeds in growth media for a week, and then half of the seeds grown were transferred to a new plate of growth media, just plain growth media, and the other half were transferred to a plate of media that was mixed with 5 millimolar of caffeine. We let the seedlings respond to this environment for 3 hours, and then we extracted RNA from the shoot tissue to measure expression levels in the samples.
So from the qPCR data, we observed a decreased relative gene expression in the caffeine treatment compared to the controls. The p-value that we acquired was 0.055, which was right on the cusp of being significant. So we believe that if we had more biological replicates, we would get a more definitive answer as to whether this decrease in the caffeine treatment is significant or not. So we were able to draw a few conclusions from all of the approaches that we undertook. Like for instance, we think that our gene is likely not involved in self-incompatibility, and that it possibly has some function in the growth and development of young seedlings, in addition to a function in the response to abiotic stressors like cold weather. We weren't able to get any information from the mutant analysis, but from the gene expression analysis, we know that expression is high in the roots and shoots, and that it follows thermocycle regulation. And we also did see altered expression with caffeine treatment.
In the end, we realized that it isn't really possible to form a hypothesis with confidence from domain and phylogenetic analysis alone for these kinds of proteins specifically. This is because of the functional divergence of G-type LEC-RLKs. So for example, here on the left, this is a phylogenetic tree that shows a large amount of G-type lectin RLKs from Arabidopsis. Keep in mind, these proteins all have the same basic domain structure, very similar sequences, but even genes in the same clade code for proteins that have wildly different functions. So had we found this paper that this figure comes from during our initial literature search about G-type LEC-RLKs, we probably would have been able to better direct and form a hypothesis. Live and learn.